Hey guys, and welcome back to Biologue. Hope you're having a great day and a great week. In this video, we're going to be looking at one of the first few topics of VCE Unit 3 Biology, which is about transcription, translation, and post-transcriptional modifications. So if you guys like this video, make sure to subscribe to Biologue, and without further ado, let's begin. Alright, looking at the next thing, which is transcription, which comes under the study design dot point steps in gene expression, including transcription. So transcription first off starts with a double-stranded DNA molecule. Now remember, in the DNA molecule you have nucleotides or bases that are joined by hydrogen bonds. So these bonds, which is the double bond between the adenine and thymine, remember adenine and thymine has a double bond. The triple bond between the guanine and the cytosine, remember guanine and cytosine has a triple bond. These double bonds and triple bonds between the nucleotides, which is the bases in the double-stranded DNA, is called a hydrogen bond. So these bonds are called hydrogen bonds. So the first step in transcription is actually to break these hydrogen bonds. And how that happens is by using an enzyme called RNA polymerase. So the first step in transcription is that RNA polymerase binds to a region on the DNA called a promoter region. Alright, so if we have these two strands connected, alright, it's not a very clear diagram, but if it looked like this, it will bind to a region on the DNA, to the promoter region on the DNA. So it binds to a region on the DNA called the promoter region, which we saw in the previous part. What is the promoter region? The promoter region is a short sequence of nucleotides to which the RNA polymerase binds to initiate transcription. So RNA polymerase binds to the promoter region of the DNA, and you will also have transcription factors which do the same exact thing. So transcription factors and RNA polymerase binds to the promoter region of the DNA, and when it binds, it weakens the hydrogen bonds between the DNA nucleotides or bases. Weakens the hydrogen bonds between the DNA nucleotides or bases connecting the two DNA strands together. So there are bases, remember the bases are ATGC. These two bases pairs will connect the two DNA strands together. So it breaks the hydrogen bond between the two bases pairs or base pairs between the two DNA strands and separates the DNA strands. So now the DNA will unzip or unwind. DNA unzips or unwinds to give you two separate DNA strands. Okay, that's the first step in transcription. The second step in transcription is that once you have your two DNA strands separated, one of the strands is called a template strand, other strand is called a coding strand. How this works is because DNA acts as an anti-parallel structure, meaning that the two strands of DNA run anti-parallelly, meaning they are anti-parallel strands. So anti-parallel strands are such that one strand of DNA will run from the 5' to 3' direction, 5' to 3' direction, and the other one will run from 3' or 3' to 5' or 5' direction. The one that runs from the 5' to 3' direction is your coding strand. Why? Because the mRNA, which is your new strand that you're going to be synthesizing, that can only be synthesized from the 5' to 3' direction. So if we know that DNA is anti-parallel and two strands run in the opposite direction, then if one strand runs from the 5' to 3' direction, let's say always your new strand, which is your mRNA strand, runs from the 5' to 3' direction, the other strand should run from the 3' to 5' direction, right? So the actual strand that we are going to be using in transcription is the one that runs from the 3' to 5' direction. And because this is the strand we are going to be using in transcription, this strand that runs from the 5' to 3 from 3' to 5' direction is called your template strand. Whereas the one that we are not going to be used in transcription, is 5' to 3', which is your coding strand. So it's a little bit confusing, yes, but the coding strand is not used in transcription, and the template strand is the strand of DNA that is used in transcription. So this is used in transcription. Now if we take a closer look onto the template strand of DNA, so if we have your template strand of DNA running from 3' to 5' direction, now the RNA polymerase brings free nucleotides from the cytoplasm into the nucleus, because remember, transcription process happens in the nucleus. So RNA polymerase brings free nucleotides. Okay, it brings free nucleotides from the nucleus, or from the cytoplasm. From the cytoplasm, if this is your cytoplasm, it brings free nucleotides, meaning free bases, from the cytoplasm into your nucleus, and the nucleus is where the transcription process actually happens. Once these free nucleotides are brought in, RNA polymerase will attach the free nucleotides onto this new strand. If we also take your so this is your DNA template strand. Alright, the one from 3' to 5' will have, let's say, if it has the sequence A, C, T, G, for example, this new strand, which is mRNA, remember the complementary base pairing rules are very important. In mRNA, then, if we had 4 bases like this runs from 5' to 3', that's very important. And now the RNA polymerase brings free nucleotides from the cytoplasm into the nucleus, but these free nucleotides are complementary to the nucleotide sequence of the template DNA strand. So if you have nucleotide sequence of the template DNA strand is A, C, T, G, then the free nucleotides that are coming from the cytoplasm into the nucleus for the mRNA synthesis is going to be complementary to these nucleotides. So if we have A, remember, in the previous part which we talked about purines and pyrimidines, we know that adenine will bind to uracil in mRNA. There is no thymine in mRNA. So adenine binds to uracil, cytosine binds to guanine as usual, thymine will bind to adenine. It's not the other way around. Adenine will always bind to uracil. Okay, adenine binds to uracil, not thymine binding to uracil, because just think of it as mRNA has no thymine whatsoever. So then if mRNA has no thymine, then thymine in the DNA will not translate to uracil, right? Or it will not translate to anything. It will translate to adenine. So always think of it as adenine to uracil and not thymine to adenine, right? It's A to T and not T to A. If you think of it as A to T, then you can easily say that in the mRNA it is A to U. Alright, so again T to A in the mRNA, and then you have G to C. Once the complementary base pairing happens, then the so actually we started with the promoter region, correct? We started with reading the promoter region over here, correct? So when we started with the promoter region, it should also end with something, correct? So now the RNA polymerase will continue to add free nucleotides onto the new mRNA strand by through complementary base pairing until it reads a termination sequence. Until it reads something called a termination sequence, which is basically a short sequence of nucleotides on the DNA that signals for the end of transcription. That signals for the RNA polymerase to stop, combining the free nucleotides together based on complementary base pairing, and to stop the process of transcription. So once it reads the termination sequence, the mRNA strand is now released. So this was your entire process of transcription. One very important thing to remember is that now, once the mRNA gets released, it is actually called a pre-mRNA molecule. Why? Because it has not undergone any post-transcriptional modifications. It has not undergone any post-transcriptional modifications, which is what we are going to be looking at in the next part. Alright, looking at the next process, which is post-transcriptional modifications or RNA processing. For this, you will need to use your number 1, the pre-mRNA strand. Now remember, the pre-mRNA strand needs to undergo RNA processing because it contains certain non-coding regions of DNA called introns. So you will have sequences like this, alright, where some of them will be coding, which is the exons, and some of them will be non-coding, which is the introns. So for example, if I label the introns with a darker color, alright, so let's say these are your non-coding regions of DNA that are not involved in translation. so they do not code for specific proteins, and these are your exons, which are coding regions of DNA that actually do code for specific amino acids and are hence involved in the protein synthesis process. So during RNA processing, we need to be able to remove the parts that do not code for any protein, so that your mRNA sequence can now be readable by a ribosome. It can actually be read properly by a ribosome to create specific amino acids and hence create specific proteins. These introns, which are non-coding regions of DNA, the non-coding regions of DNA need to be removed, and the exons which are expressed, okay, exons are actually expressed sequences of DNA that are expressed to form specific proteins or specific amino acids which are then joined together to form specific proteins. These ones are your coding regions of DNA, okay, that actually are involved in the translation process. So introns are non-coding regions of DNA that need to be removed from the pre-mRNA during RNA processing in order to form mature mRNA, which is a mRNA type that is readable, that actually can be read by the ribosome in order to make specific amino acids needed to make specific proteins. So once this happens, basically the introns will then get removed. These introns will be removed through spliceosomes, okay, through something called spliceosomes. Once these introns are then removed, all you will have is your combination of exons. All right, all you will have is your combinations of exons. These exons now will then be spliced together. They'll be joined together or they'll be spliced together by the same spliceosome. So the spliceosomes here will splice together the exons. Once the exons get spliced together, now at this stage we call this molecule a mature mRNA molecule, because it has undergone post-transcriptional modifications of the removal of the introns, which are non-coding regions of DNA. So that's pretty much it for RNA processing. If we look at again the distinction between prokaryotes and eukaryotes, remember prokaryotes only have exons, they do not have introns, so we do not need RNA processing or post-transcriptional modifications to happen in prokaryotes, but eukaryotes will have introns. So you need to be able to remove those introns in order to make this mRNA sequence readable by the ribosome for translation. So it will have RNA processing or post-transcriptional modifications. Another important concept is the concept of alternative splicing. Now alternative splicing is the reason why sometimes we can say that there are many different types of proteins produced in a cell, right? Alternative splicing actually allows us to produce different types of proteins in a cell, okay? It allows us to produce many different types of proteins even though we have only one mRNA strand, okay? We have the same mature mRNA strand. Okay, so basically in alternative splicing, sometimes not only the introns but also some exons can be spliced, or some exons can also be removed. So alternative splicing involves of removal of certain exons so that in the mature mRNA sequence there are different combinations or different orders of exons in them. So then these different orders of exons can code for specific amino acids and hence can code for different proteins. So basically if you splice out different exons, then different exon combinations or orders will be there in the mature mRNA, or orders will be there in your mature mRNA. And so this mature mRNA, once it gets translated, it can code for specific proteins, different specific amino acids, which can then code for different proteins. So overall, this is the process of RNA processing, or pretty much alternative splicing or gene splicing basically. All right, moving on to the next concept, which is translation. Now for translation we need to basically know each and every step in translation. So now we have our formation of mature mRNA. The mature mRNA, remember, as we said, it contains a 5' end and a 3' end. this 5' end number 1, the ribosome will read the 5' end of the mature mRNA strand. So let's write the step for reach the 5' end of the mature mRNA strand by number 1 reading the start codon, which is basically AUG. Once the ribosome reaches the start codon, translation is initiated. Okay, translation is initiated by the tRNA molecule bringing a complementary anticodon. It brings an anticodon into the ribosome. Okay, this anticodon is complementary. This anticodon is basically complementary to the codon of the mRNA sequence. Then the tRNA will bring a specific amino acid, based on the codon of the mRNA. Okay, so remember the amino acid is actually coded for the mRNA's codon, not the anticodon. So it brings a specific amino acid based or coded for by the mRNA's codon. And then the fourth process is that once the tRNA brings the amino acids are then joined by peptide bonds. They are joined by peptide bonds, okay, through condensation reactions, which we talked about in the previous part. So their amino acids are joined together by peptide bonds through condensation reactions at the P site of the ribosome. P site meaning polypeptide site, okay, at the P site of the ribosome. And the last step is that once it is joined at the P site of the ribosome, the amino acids then form a growing polypeptide chain. They will then form a growing polypeptide chain. After this, once the ribosome continues to read along the mRNA strand, when the ribosome reads the stop codon, remember stop codon and start codons are important because they stop and start, initiate and terminate the translation process. So once it reaches the stop codon, translation gets terminated. Okay, translation is terminated. How is it terminated? It is terminated because your growing polypeptide chain, gets released. Okay, this thing will get released. Alright, so that is basically your entire process of translation. Okay, yeah, so next thing is to look at the differences. Now that we've actually covered transcription, RNA processing and translation, we need to look at the differences between the mRNA, between the RNA types, which is basically mRNA, tRNA, rRNA. Okay, so mRNA. Okay, so mRNA basically is it's the molecule that brings up genetic information. So it brings the genetic code. Okay, it brings the genetic code for translation or synthesis of proteins. Of specific proteins from the nucleus into the ribosome. Next thing was tRNA. tRNA brings a complementary anticodon. So basically a complementary anticodon to the codon of the mRNA. And based on this, it will actually bring a specific amino acid coded for the mRNA. So amino acid coded for by the mRNA's codon. into the ribosome. Okay, basically it carries a specific amino acid. That's the main part. Carries a specific amino acid, and then these amino acids are then joined by peptide bonds. Through condensation reactions. Okay, so last one was rRNA, which is ribosomal RNA. Name itself suggests that it is the structural component that makes up a ribosome. So ribosomes are basically made of rRNA and protein. So yeah, that's all for transcription, post-transcriptional modifications and translation. Thanks for watching, guys. If you guys like this video, make sure to like, share and subscribe to Biologue. I have a lot more VCE Unit 3 and 4 content and topic videos coming up for you guys. So make sure you hit that subscribe button, and I will see you guys in my next video. Have a great day.